daughter cell that has the same number of genetic content as the parent cell, then it means that at each stage or at each level of reproduction, where both, sexual reproduction now, where both parents will have to contribute genetic materials, then there will be a doubling of the genetic material possessed by the offspring formed, the zygote formed. All right. So in order to remove, in order to stop this, there is a type of division that would have to give rise to half the daughter cells that are present in the parent. And this kind of cell division is called meiotic division or meiosis. All right. Remember in my last video of genetics where we introduced you to the 100 terms in genetics, I did tell you that a good knowledge of the origin of the words in genetics will allow you to understand the concepts very well. All right. So meiosis basically is coined from a Greek word which means lessening or reducing. Lessening or reducing. Now, the fundamentality of this process is such that we create a daughter cell that has half the chromosomal number of the parent, so that at each stage of fusion we can have what is called a diploid cell. Now, this meiotic division has some peculiarities, part of which is that it produces what is called haploid daughter cells. Haploid. Now, haploid, the word haploid is gotten from the Greek word haplous, which means single. And then oid is an English word, an English suffix. Alright. So haplous means single. So meaning that it's going to produce daughter cells that have a single unit of the genetic pairs that came from the parents. So meaning that after fusion of both gametes, which are both aplontic, then there is a production of the zygote, which is diplontic. Alright. So basically, I'm going to run through all there is that happens in meiosis. There are two types of divisions which I'm going to go back to that happens in meiosis, and they are called, the first meiotic division is called meiotic division 1. Now, that division is called the reductional division. And the second division, as meiotic division 2, is called the equational division. Now, generally, meiosis is termed a reduction division simply because it reduces the number of chromosomes in the parent, all right, by half. So in the reductional division, that is meiosis 1, we always produce half of what we started with, and of course, in the equational division, there are reasons why the reduction division is different from the equational division. Now, but we named the process after the first division because that is basically what is happening throughout the process. There is always a reduction at each stage. Alright, basically, what happens is split into 4 processes. The meiosis is split into 4 processes or phases. 4 processes or phases. And the phases they are abbreviated as PMAT. Alright? Remember in your basic biology that PMAT stands for prophase, metaphase, anaphase, and the telophase. Alright. So now what happens generally is, I'm going to give a quick summary of what happens in meiosis. What happens is that there is first a thickening or yeah, of course, a thickening, a winding up or coiling of the chromosome so that they become prominent in the cell. And then thereafter, in meiosis, what happens next is there is synapses. The chromosomes synapse, and the synapses happens between the process is called synapses. And when chromosomes synapse, it is homologous chromosomes that actually synapse. Now, when after synapsing, they exchange genetic pair through a process that is called recombination or crossing over. Now, after the recombination, we have then they migrate to the spindle fiber is formed by the centriole, and the centriole, which has already moved to two polar ends of the cell, and of course the chromosomes, which are now bivalent. When they synapse, they form what is called bivalent, and after which they now move apart, and when they separate, they already exchange genetic components in the recombination process, and after which they now separate to both ends of the chromosomes. All right, and then a nucleus encapsulates them, and then there is a splitting of the cell. So that is a summary of what happens. But let's go into it in a better detail. All right, the first is prophase. Now, in prophase, excuse me, let me get a duster. All right, we are going to be starting with prophase. Now, prophase, remember the word pro. So this is prophase. Remember the word pro means like to begin, like we want to begin, like pro and pre. They kind of mean the same thing. That pro is to, at the start of something. So how meiosis starts is that the chromosomes begin to become more prominent, more apparent in the cell by thickening. They coil up and shorten and thicken, so they become more prominent. So meaning that basically before meiosis begins, the cell basically contains chromatin strands that is on one chromosome. Alright, so in that video I discussed the difference between chromatid and chromatin and chromosome, so you should know when to use each one. Alright, so they become thickened basically, and in this same prophase you begin to see that the centrosome, which is the portion of the cell where the centriole lies, they begin to separate apart, and then the centriole begin to move to the other end of the cell to form two poles. Now, so to remember this, just try and remember, to remember what happens here, just take the mnemonic, the mnemonic prepare. So remember that the prophase means the preparatory phase. Now the second phase is metaphase. Now one thing I forgot to mention here is the fact that during this preparatory phase, one other thing that happens, apart from this central moving here, is the fact that these chromosomes begin to pair up. Homologous chromosomes. That is, chromosomes that look alike, that have the same genetic composition, begin to pair up. And when they pair up, they form what is called bivalence. Now, in the full video here of meiosis, I did explain that bivalent have four arms, which are 1, 2, 3, and 4. 1, 2, 3, and 4. And those four arms are called tetrads. Alright? So now, when they get to the metaphase phase, think of metaphase as middle. So what happens is that all of them are going to align themselves to the middle of this spindle fiber, which are called kinetochores. Now, they align themselves to this middle, and they form at this center. So they align at the center with their centromeres. This portion is called the centromere. So they align themselves at this portion, and they are able to, stay in the equatorial region of this spindle fiber. like that. So then the next phase that happens is the anaphase. Now keep in mind that this prophase is the fastest, the slowest phase of meiotic cell division, while anaphase is the fastest phase. Now what happens here is basically that you begin to see these chromosomes begin to detach or like draw back to the different poles, the separate poles. Now bear in mind that here it is not chromatids, sister chromatids, that actually divides or separates. It is non-sister chromosomes. Now they are non-sister chromosomes because by the time they exchange the genetic information here, these portions where they exchange genetic information, you remember I called it chiasmata. Chiasmata is the plural and chiasma is the singular. So these portions where they exchange genetic information, where crossing over occur, makes them become non-sister chromatids. Why? Because the genetic information from parents has mixed with the one from the mother, so that when they separate, they bear different components of what they each had initially. So they separate, that is anaphase. Think of anaphase as a way. So like they move away from each other. Now, the next thing that happens, and this is prophase 1, this is metaphase 1, this is anaphase 1. Now the next thing that happens is what we call telophase 1. Now in telophase 1, what happens is basically that at telophase 1, those non-sister chromosomes already have moved to the opposite ends of the poles. Now having gotten here, a new cell nuclear membrane begins to encapsulate them, and then this cell wall, cell membrane also begins to invaginate. And once it invaginates, it tears apart. Think of telophase as tear, and then it's divides the cell into basically two daughter cells. And with the chromosomes in there. Now notice, there are chromosomes that we have here. Then we proceed to the next phase of division. Alright, usually in some animals it is immediate, and in some others there is what we call a phase where they will lag a bit before they continue to the second meiotic division. Now in the second meiotic division, alright, in meiosis 2, the process is almost the same, only that there is no need for thickening of the chromosomes because they are already thickened here. The only thing that happens again is that the centriole begin to move again to the opposite poles here, and the homologous chromosomes are already paired here. You see that there are chromosomes here. Those are the non-sister chromosomes that are actually separated in meiosis 1. Now they are here, and that is prophase 2. Now what happens next is the fact that they move to the equatorial region of stay in the middle or the center of the spindle fiber or kinetochores formed by the centrioles. Alright? Now the next thing that happens is that, so you remember I said think of this as preparation, another preparation, the preparation for the second division, M for middle, so they align themselves at the center, and here what happens is that they begin to move away. That is why it's called anaphase. Remember the word ana, I did tell you that ana means up, so they move up to that cell, move up to here, to this central, and move up to this central, this portion here. So and notice that whenever they are moving, even in that first level of anaphase, that's anaphase 1 of meiosis, the movement of that chromosome is led by the centromeric region. So this centromeric region leads the movement. It always leads the movement because, of course, that is where the kinetochores are attached to. Okay, so then So think of this as away. They move away. And so notice, in the first phase, non-sister chromosomes moved apart. But here, non-sister, chromatids now move apart. So here, I told you that in the first video, I did tell you that this means when they form a bivalent, it is called they form a bivalent they form a bivalent that each arm is called a tetrad. So because the four arms are called tetrads. Okay, so here when they separate, they form a dyad. So this is a dyad when they are separated in the first place. Now when they separate finally here, they form a monad. So remember mono, which means one, di meaning two. So the tetrad becomes broken down to monad. So when they move apart, of course, the next thing that happens is think of telophase 2 as tearing apart telophase. So this separates the nucleus, nuclear membrane begins to form around the new developed chromosomes on this side and of course on this other side, and that is the end of the division. So listen up, you notice that the same thing. Now it doesn't mean that it is two daughter cells that are formed there. In fact, there are four daughter cells that are formed. Why? Because we divide it into two there. So those other two, one of it will divide into these two, and another one will divide into these two, giving rise to four daughter cells, four aplontic daughter cells. Right? So that is what happens, the brief detail of what happens in meiosis. Alright? But if you want to really understand each of the stages, prophets, do well to check the link in the description. There's another video there which explains totally everything almost everything that happens in meiosis. Thank you very much for sticking to the end of this video. Do well to subscribe to the channel, and God bless you. Amen.